Heteroduplex analysis. This is a special technique used to detect errors in genes. In heteroduplex analysis, we use a sample which contains two types of DNA. DNA with mismatch pairs known as mutant DNA and DNA without mismatch pairs known as normal DNA. Now we use a method called PCR to make copies of the DNA sample. PCR can amplify both normal and mutant DNA sequences present in the sample. In the next step, we get things hot. We raise up the temperature to denature the DNA strands. After strand separation, we cool things down again by lowering the temperature. Now here comes the magical part. When we drag down the temperature, the separated strands pair up randomly. Some DNA strands stick back together perfectly. They are called homoduplex. While some have mismatch errors in them, we call them heteroduplex. We now use special enzymes called single-strand specific nucleases to cleave the mismatch pairs. These enzymes target the mutant DNA strand and cleave the mismatch part. Finally, we use gel electrophoresis to visualize the cleaved DNA fragments, which helps identify the presence of mutations. Heteroduplex analysis checks for errors in the DNA. By detecting these mismatched base pairs, heteroduplex analysis helps scientists understand genetic diseases and potentially develop cures.